Welcome to the epigenetic part of the lecture again. So last lecture we had a visiting or a guest lecture who discussed DNA methylation, but I want to give a very quick introduction of epigenetics. So people were started, or at least a lot of people were interested in epigenetics because they see these heritable changes in gene function that occur without the change in the DNA sequence. For example, from one cell to the daughter cell after differentiation, a blood lineage cell will know that it's already blood lineage instead of being a brain or a muscle cell. How does the cell remember this, right? If it's all encoded in the DNA sequence. And also when people are doing ChIP-seq for the transcription factor in different tissues, such as androgen receptor binding, say in prostate versus other liver or other tissues, you would see that even though transcription factor motif influence the transcription factor binding in the different cells, the factor will bind to different locations, and not all the motif occurrences were bound by the factor. And also, even with the transcription factor binding, it doesn't necessarily regulate the nearby gene. Sometimes it just parks there without influencing the nearby gene. And so what influences these? And that's kind of how a lot of people are interested in epigenetics. If we only look at the DNA sequences, kind of pretty much all the cells and also all the individuals have the same DNA, but this is actually packaged in different levels of the chromatin. The very basic unit of the chromatin is called a nucleosome. Basically, there are 8 histone proteins that's kind of forming a nucleosome, and then there's 146 nucleotide that's DNA wrapped around this 8 histone proteins in 2 circles. And so you can see here, the blue here are 8 histone proteins, and the red is the DNA wrapped around in 2 circles and then move on. And then there's another nucleosome and another nucleosome. And this can then be stacked up together to form this 30 nanometer fiber. And then if you zoom out even further, you'll see something like this. This can be even further packaged. You can imagine this as a rubber band. You keep twisting, and then some regions become really tight, whereas other regions are still pretty relaxed. During the metaphase of the cell cycle, the chromosome really gets very compacted, and at that time, no gene is transcribed. The transcription activities are completely silenced. But during the regular cell cycle, in the non-division phases, the cell will be kind of having more relaxed chromatin, and the really open chromatin regions will be active for transcriptional activity. And so understanding the epigenetic the cell will tell us which locations are more compact versus more open, which will dictate what kind of genes are going to be transcribed. And so in the last lecture, we discussed DNA methylation, which are modifications on the DNA directly, but you can see there are different levels of epigenetics, and the very first level is nucleosome, and the positioning of the nucleosome also matters. So this is another view of nucleosome. You can see here this DNA double helix, and then it wraps around these 8 different histones in 2 circles, and then it will move on and then wrap around another 8 histones. And the basic unit of the chromatin is this nucleosome, which is 8 histones and 146 base pair of DNA. The position of the nucleosome is important in that very often transcription factors would prefer to bind to the nucleosome-free regions like this region, whereas if the region is occupied or the DNA is occupied by the histones to form a nucleosome, many transcription factors would not be able to come and bind to these regions. And so in order to really map the nucleosomes on the chromatin, there are experiments that can be done. We digest the chromatin with an enzyme called microcalcanuclease, or MNase. And this MNase basically will cut, it will have an easier time to cut in these open regions, nucleosome-free regions. And then you will be able to get just the nucleosomal DNA. And then you isolate the DNA and sequence them out, very often now from the two ends, which the nucleosome is right in the middle, and the two ends are the two ends here and there, right? So using this approach, we can sequence out the nucleosome and see where they are. This is an example from a 2009 review. At that time, most people only sequence one end of the nucleosome. At that time, paired end sequencing wasn't so popular yet. But again, this is similar to like a transcription factor binding. You will see two peaks in here, which usually is to the left and the right of this nucleosome. And if you shift the sequence to the middle, that will give you a peak shape. And here we know roughly how much to shift because the nucleosome is 146, so you only shift about half of this 73 nucleotides. Then you will see the nucleosome positions in the genome. In the reality is that if you can imagine, usually this experiment is done in millions of cells, and in the different cells, the nucleosome, it's like a pearl on a necklace or beads on the string. They can slide around. And so when you sequence this, you don't always see a lot of peaks because when the nucleosome are not well positioned, the nucleosome can kind of freely move around across the genome. And so the signal is kind of more even. But occasionally, you would see a very well-positioned nucleosome, which means across millions of cells or many, many cells, this nucleosome is always at this position. And then you will see a very well, peak kind of situation. And so there are different factors that influence nucleosome positioning. The first is DNA sequence. This people call it like a cis element or DNA influence, which is like this. If you look across the genome for AA versus TT or TA sequence, and then compare it to the locations where you have either CC, GG, or CG sequence in the genome, across across eukaryote genomes, you would see something like this, starting from yeast, then many animal species here will have this 10 base pair periodicity, which is the percentage of AATTTA fractions. It's a very small fluctuation, but it's kind of consistent across the a genome. If you just look at any region, you will see this kind of weak 10 base pair periodicity. This is because the DNA that's wrapped around the nucleosome, the DNA have a self-rotation in that double helix. You can see here, as the DNA wrap around the 8 histone, the double strand helix also have the self twisting in here. And interestingly, the GC sequence actually prefer to interact with the histone proteins inside, whereas the AT sequence does not like to face the histone proteins. And so if there are this 10 base pair periodicity, what happens is that usually the AT will always be facing outside, whereas the GC will be facing inside of the nucleosome. And because the double helix is a 10 base has a 10 base pair, each round of the double helix, the self twisting is 10 pair of 10 pair of nucleotides stacking on top of each other. And so basically the 10 base pair enrichment of the AT sequence will make sure that or at least will favor an AT sequence facing outside and GC sequencing face inside. And this phenomenon is only observed in eukaryotes when there are nucleosomes. For example, in bacteria and virus, there is no nucleosome, there's no histones, and therefore you don't observe this type of 10 base pair periodicity. And so you can imagine when there is the DNA that can either be here or here, right there, like within the 10 base pair region, there is a favored position when the nucleosome can fit snugly when the GC are aligned inside. But this actually have a very small influence. The bigger influence is really transcription factor binding. So as we mentioned, if you sequence nucleosome across the genome, in many locations, what you will see is like this. The nucleosome are not very well positioned. However, in some regions, in this case, you will see a very strong nucleosome-free region. The reason there's no nucleosome here is because the polymerase is a very strong transcription factor, and it recruits many other transcription factors and cofactors to bind to this region, and this will really squeeze out the transcription, squeeze out the nucleosome. And this region suddenly has no nucleosome, and because of this, the adjacent pearls on this necklace will also be kind of slightly better positioned. And then the next one will be better positioned. But gradually, as you go inside the genes, eventually, this is like it's like a track field. Everybody start from here, and then initially they are still synced, but gradually they will become out of sync. And you can see here, you lose the positioning of the nucleosome. And so you can see here, nucleosome positioning can influence transcription factor binding, but they are also influenced by transcription factors. For polymerase 2, which has a very strong propensity to bind there, they can actually squeeze out the nucleosome and force the nearby nucleosome to be better positioned as well, although this can gradually be out of sync over distance. And so this actually are translational nucleosome positioning, and they really are the more important factors influencing nucleosome positioning. And also these type of nucleosome positioning are better at predicting where transcription factor can bind. Okay, so that's the first part. With nucleosome as the basic unit, you can see here, each of these nucleosome on the histones, they also have little tails that's kind of dangling outside. And these tails can be modified. And depending on the different modification, if you imagine strings on, beads on a string or pearl necklace, you can imagine some of the pearls have a little flag. And because the eukaryote genome, especially you can imagine the human genome is huge. And for a transcription factor to be expressed and going to the nucleus, it's really quite blinding. It has to really quickly find where to bind out of 3 billion locations. And imagine having to go through a lot of locations and trying to see where this transcription factor motif exists so that the transcription factor can bind there. It takes a lot of trial and error. So histone modification or epigenetic in general, you can imagine are beacons in the genome which tell transcription factors or other factors where to go first. And so very interestingly, this histone modification is a very, very powerful beacon in the genome that tell the transcription factors where to go. And as we mentioned, in the nucleosome, there's like 2 rounds of DNA, and there are 8 different histones. And there are 2 histones called H2A here and 2 H2B, and 2 histones H3 and 2 histones H4. And they are kind of 4 together and they stack on top of each other. Actually, I don't think this figure is accurate. It should be the 4 with 1 circle and another 4 with the circle, not the other this way. Anyway, and you can see here, this is the histone tails that comes out of this, and these can be modified. And based on the different modification, you can also specifically enrich for locations in the genome that has the histone modification. And this can be done with ChIP-seq. So you can imagine if we fragment the chromatin, remember in ChIP-seq, we fragment the chromatin into, say, a couple hundred base pair fragments. And normally for transcription factor binding, you just use an antibody against the transcription factor. But once the chromatin is fragmented, we know if the histone tail is modified, there are antibodies against modified histones. You can use antibody to kind of collect all the pearls that have the little flag and then isolate the DNA that's wrapped around that nucleosome and sequence those out, then you will know which location in the genome the histones are modified. And so this is called histone mark ChIP-seq. And so in the genome, these little marks can be added by readers or readers. They can also be removed by erasers and also be read by histone mark readers. And so depending on the different location, and there are also different marks. And this is just one example. There are modifications also on H2, also H4, but this is just one example of the histone H3. Position 1 is on the is at the end in here, and as you go inside, this will be more like a position 79. And so you can see here, most of the modification happen on the arginine and lysine. R is arginine, K is lysine, or threonine T, and also serine on P, and also maybe on P locations. And so depending on the different locations, for example, you would very often see something called H3 lysine 4 trimethylation. It means that on this tail at this fourth position, because there is a lysine, you can see there are many different modifications that can happen. And in this position, if there are 3 methyl marks, that's called a H3K4 trimethylation. If there is only one, so basically on the carbon of this lysine, if there is one methyl mark, that's monomethylation, because the carbon, one end is attached to the remaining amino acid, and then the carbon has 3 hydrogens. If one hydrogen is replaced with a methyl, that's monomethylation, Me1. 2-methylation will be methyl 2, and trimethylation will be Me3. So all 3, hydrogen bond are being replaced by methyl groups. So that's H3K4 trimethylation. Another one that's quite popular is actually not really indicated in here, but on this lysine 27 position, it can be methylated, but also can be acetylated. And that would be called an H3K4 acetylation. And so this will help you know how to read different histone marks. And in here, you can see there are many, many different names in here. These are the readers or writers or erasers. You can see if there is a stop sign in here, that means it's a lysine demethylase. Whereas if it's black, it means it's a writer in here. And also in this case, it's a H3 lysine 36 writer is called lysine methyltransferase at this location in here, which will add, you can see it's a very complicated mechanism. There are many, many writers, many, many erasers, and many, many readers, which add and remove these histone marks. And there are also something called a chromatin remodeler. So remodeler, place the nucleosome into different locations. They kind of help position them or help slide the nucleosome along the DNA. And writers add the mark, erasers remove the marks. Readers can recognize the marks, and usually by the reader, after the recruitment or after the recognition, it will try to recruit other factors such as transcription factors and also help recruit additional writers and erasers. And so epigenetic in the cell is in a dynamic process, right? So these kind of writing and removal and reading and recruitment really happens all the time. And when you do a ChIP-seq, you just capture a snapshot of what's happening in millions of cells. And so there are some clarifications. When we talk about DNA, it's really the naked DNA double helix without any higher or without any structure. But when we talk about chromatin, it means that it's DNA in its compaction structure in the cell, including nucleosome, including the higher order compaction, and also the other modifications. Nucleosome is the basic unit of the chromatin. It's 146 base pair DNA wrapped 2 rounds around 8 histone proteins. And for regular transcription factor ChIP-seq, you use sonication to fragment the DNA. You use the antibody against the transcription factor that binds to the DNA. For histone-marked ChIP-seq, you can use sonication, or you can use micrococcal nucleus. Basically, you cut on all the nucleosome-free regions, and you collect all the nucleosome like pearls with the DNA still around it. And the antibody is against the modification on the histones. And if you look at also the nucleosome sequencing here, you sequence all the nucleosomes, and you can see here only occasionally the nucleosomes are well positioned, but in many other locations it's kind of randomly spaced. Whereas when you look at histone modifications, in this case, if it's micrococcal nucleus treated, you don't really sequence all the nucleosomes. You only look at nucleosomes that are modified, having specific modification. And so this is kind of a cheaper experiment. Instead of collecting all the pearls, say you only collect the red pearls or pearls with a red flag. And in here, you can see the signal here is much, much stronger. You still see the same positioning of the nucleosome. And you can see here, this one has a very strong, like a very strong flag. And the adjacent one is kind of weaker, because usually when the histone mark writers go there in the right position, it will add the mark. And then, like nearby things get like modified a little bit, but they're not the main players. So the strong modifications actually indicate strong signals, and that they send signals to, say, when a transcription factor is available in the nucleus, it will look for locations where these marks are, and it can find the locations much, much faster. And so these are much higher resolution. It has better signal to noise. The difficulty is there are many different marks. And so every histone mark ChIP-seq is a separate experiment. In terms of the function of the histone marks, as we mentioned, these are like beacons in the genome. They kind of wave their flags to tell transcription factors or polymerase where to go. And depending on the different histone marks, you can use this, now people are using this as a tool to understand what genes are important, which locations are important. And in some sense, experimentally, scientists are also taking advantage of these beacons in the genome to help us understand how genes are regulated. And so if you look at epigenetic profiles across the genome, there could be some regions that have bisulfite sequencing, and across the genome, you might see this type of signals. If you look at H3K27 acetylation, you can see here, it's much weaker across the genome, but then in certain regions, it's suddenly stronger. And remember, because DNA methylation is normally silencing gene transcription activities. And so interestingly, in this case, when DNA methylation signal is much, much weaker, H3K27 acetylation is active or is stronger. K27 acetylation actually tells us which regions of the genome are really, really open and active. Whereas, there are other marks, K4, mono and trimethylation, that you can see they also have signals across the genome and kind of more focal on some of these active regions. So you can see here, different histone modification have a different genome-wide distribution. And here is the gene annotation. And over the years, what people have learned are the following. These are some basic knowledge. H3K36 trimethylation is a mark that polymerase leave as it transcribe a gene. And this helps polymerase to know where to start the transcription and where to slow down. For example, K36 trimethylation are usually stronger on the exons, it is slightly lower in the introns. And also as polymerase go across the gene towards the later part of the gene, K36 signal is stronger. This is because I'm actually suspecting that polymerase, once it starts transcription, it will start faster. And then as it go later in the transcription, it actually moves slightly. I suspect it's actually moving slightly slower. And when there are introns, if there is any transcription that is a mistake, from the DNA as a template to make the RNA, if there are mistakes, no big deal. But when it's exon, polymerase need to know, okay, I need to slow down because the transcript actually is important for later making the protein. Therefore, the transcription is a little slower. The K36 trimethylation signal kind of indicate to PO2, go a little slower. This region is more important. And so PO2 elongation is being guided by H3K36 trimethylation. But as polymerase go, polymerase recruits the methyltransferase that adds the K36 trimethylation. So initially, without guidance, PO2 probably go quite slowly because it has to decide, where to, how fast to go, and there's no guidance. But then with every round of transcription, PO2 will recruit the K36 trimethylation enzyme to add the marks there. Then in the next round, it will know how to move, like which part to go faster, which part to go slower. But as it moves, it continues to recruit the methyltransferase to add marks to this to help the next round to be more smooth, and so on. H3K79 trimethylation is stronger in the first exon and the first intron, and then the later exons or gene bodies are much, much weaker. H3K4 trimethylation is a very strong mark near the beginning of the gene, and it tells the cell where a gene has the promise to be activated. You can imagine this kind of is like a door that's only slightly open just to tell the cell this gene has the promise to be turned on. In the enhancer regions or the transcription factor binding site, if there are H3K4 monomethylation mark, it means that this region has the promise to be turned on, even though, or be bound by the transcription factor, even though that region may not be heavily bound by the transcription factor yet. There are studies showing that in a lot of cells, once the lineage of the cell is determined, there are small, like low levels of the relevant transcription factors that are expressed, and very small level of these transcription factors can bind to occasional locations in the genome. They prime those enhancers to have the H3K4 monomethylation mark. So these are kind of, doors only slightly open, just to show that these regions can potentially be turned on. But then when the outside stimulus is ready, or when the transcription factor really turn on a lot, then they would go to those, like slightly open locations first, and then check, does it have the motif, and then will bind strongly. And once they bind very strongly, the histone marks will become much, much stronger. And then this region will really get turned on. And then the promoters for those genes that have the trimethylation mark, initially they will just, they have the promise to be turned on, but then once the transcription factor binds to the nearby regions, that really kicks off the polymerase from actively transcribing the gene. And there are some marks that both the promoters and enhancers, such as H3K4, dimethylation, and all the acetylation marks. Actually, acetylation marks in general indicated activation. This door is now totally open. And so a region that's really bound by a lot of transcription factors on the enhancer region will have K27 acetylation mark. And the promoters that's actively transcribing the gene on the promoter regions will also have very strong K27 acetylation signals. And so you can see here, the different marks really show a different thing, and they also help to guide the transcription factors. There are also repressive marks in the genome. For example, K27 trimethylation. You can see in a lot of cells that it binds to the CpG island near the promoters. This especially happened during a stem cell differentiation. That actually indicate this door is kind of inaccessible, is closed. And also H3K9 trimethylation. This happens in the repeat regions of the genome, which is probably half of the genome. And in the initial stage, when the region needs to be silenced, it will use K9 trimethylation. But then for even longer term, long-term suppression, you can also use DNA methylation, which is, not only door is closed, you add a log to it, like a big log in here, or use a board to board it up this region. Then you say, okay, this region is really off limit in those repeats. Yeah, so even though the overall resolution of HistoMark ChIP-seq is slightly lower than the TF ChIP-seq, you can see here, we can already use the marks to tell us a lot about what is happening in the genome, which regions are interesting, and use it to understand gene regulation. So we mentioned here, very often nucleosomes, DNAs that's wrapped around the nucleosome are like preventing transcription factor from binding. There are two exceptions. One is if you have multiple transcription factors that kind of form a complex and each have some DNA specificity, by working together cooperatively, they can eventually squeeze out this nucleosome and can basically kick the pearl out of the necklace or the string. There are also some transcription factors called pioneering factors. They sometimes even prefer to bind to these nucleosomal DNA regions. And the reason they are called pioneering factor is once they go there, they can kick out the nucleosome and they can then help additional transcription factors or other transcription factors to bind to this region. And then that will help transcription of the nearby genes. So you can see here, the transcription factor and epigenetics is kind of a chicken egg problem. Initially, when the transcription factors are expressed in the cell, it's quite blinding. It has to test many locations, but very often it goes to the regions that already have histone marks. So usually when this pioneering factor binds in here, it kicks out the nucleosome, but it will also recruit other histone mark readers to help mark the nearby nucleosomes. And once these nearby nucleosomes are marked better, other transcription factors will go there very, very quickly because they know there is a nucleosome-free regions that can potentially be bound. And so you can see the histone marks on the nearby location really help, like flag the locations of interest. And so this is really kind of a chicken egg problem. Epigenetics help transcription factors to find where to go first. But those transcription factors, by working together or through these pioneering factors, can also kick out nucleosomes and add more marks to the nearby nucleosome. So that actually can help understand this. And so, for example, a lot of people use HistoMark ChIP-seq, especially H3K4 monomethylation and H3K27 acetylation ChIP-seq, to identify transcription factors that are important in the cell. And so usually what they do is they do this ChIP-seq, and in terms of analysis, the very first thing you do is you get FASTQ file, and you can analyze it in a similar way as when you do ChIP-seq. You do the read mapping using BWA, You can call the peaks. You can also call the differential peaks between, different conditions using max. You can do the standard QC to make sure this experiment actually worked. And then we know that if it's a K4 monomethylation or K27 acetylation ChIP-seq, it's marking those enhancer locations. And from there, from the recent homework, you can probably see you can do motif search in those peaks. Maybe that can help you at least figure out what family of transcription factors might be important in those potential binding sites or the histomark peaks locations. And then using the K27 acetylation at the read level. So actually, if you look at if you remember the transcription factor binding lecture, we talk about regulatory potential, which is we weigh the ChIP-seq signals from nearby regions, say within 100 kb with an exponential decay to decide, how much a binding site will have effect on the nearby gene. And we can do the same thing to calculate regulatory potential on the K27 acetylation to calculate the effect on nearby gene. And interestingly, we found that using this approach, you get a pretty good idea which genes are activated and which genes are not actively transcribed, which means that if on this gene, there are a lot of K27 acetylation signal, this gene is very likely to be highly transcribed. And also if there are nearby signals of K27 acetylation, that also indicate that there are strong transcription factors binding to the nearby regions and they help to promote the expression of this gene. And so using this approach, you can sometimes guess which genes are active. And so if they are, say, if there is a GATA family of transcription factors, there's GATA 1, 2, 3, 4, 5, 6, they all recognize the same motif. From motif search, you might find the whole family to be potentially interesting. But then if you use the K27 acetylation to calculate this regulatory potential, you might see that, oh, in this cell, actually it's the GATA3 that's activated, but not GATA 1, 2, 4, 5, 6. And so that can help you inform on the gene expression. You can also use the K27 acetylation signals in the nearby transcription factor binding sites to weigh the peaks. So we mentioned that previously in transcription factor ChIP-seq, whether the peak is a 10-fold versus 20-fold, it probably tell you that the transcription factor is binding stronger to the region. But in terms of regulating nearby gene expression, the 20-fold peaks don't necessarily have a stronger effect on the gene expression than the 10-fold peaks. At least from 20 years of people modeling transcription factor binding, there is a difference between the signal from the noise. Like if you are calling a peak with only 1.2-fold, that maybe they don't really have effect. But once the peak, TF binding is at 5-fold, whether it's 10-fold or 20-fold or even 50-fold, in terms of the TF ChIP-seq signal, it's not known to be really, having a stronger effect on the gene expression nearby. Interestingly, if you look at that particular peaks, TF binding location, and look at, how much K27 acetylation signal is in this location, it does have an effect. For example, if you have two peaks nearby, and one peak has like 50-fold K27 acetylation signal, and another peak only have 5-fold K27 acetylation signal, we will know that the 50-fold K27 peak, that binding site have a stronger effect on this nearby gene than the 5-fold one. And so you can use the K27 acetylation ChIP-seq signal, probably at the read level, you can do either a square root or a log of the K27 reads in that peak to weigh the transcription factor binding peak and see whether they have an effect on nearby genes. I think that actually usually give you better result. Another way, besides motif finding to find the relevant TF is you can just overlap the TF ChIP-seq data in the public, because there are like tens of thousands of ChIP-seq data in the public. And if you just do a peak significant overlap with your K27 acetylation ChIP-seq or K4 monomethylation ChIP-seq peaks, and ask which public ChIP-seq data have significant overlap with my histomark ChIP-seq, you can actually guess which transcription factors are important are important for this particular cell situation. The nice thing about using a histomark is for transcription factors, we have to know what factor is important for the cell. For example, in homework 3, you see that we know that androgen receptor is important in prostate cancer. Therefore, we do a androgen receptor ChIP-seq. But hopefully from this, you will find, oh, there are other transcription factors that might be important. But what if we don't know what transcription factor is important for, say, brain cancer? What you can do is you get the brain cancer tissue, and then you just do a K27 acetylation ChIP-seq, and based on the peaks, and you overlap with all the known transcription factors or look at the motifs, it can tell you what transcription factor is really important for brain cancer without prior knowledge. And that's actually the value of doing a histomark, because it not just captures one transcription factor, it captures many, many transcription factors in this cell. And so using the Cystron toolkit, which you will see from the homework, you will see that using the histomarks, you can figure out what transcription factors are important, and you can also use differential peaks to do this overlap. And so this kind of utilizes a Cystron database, which collects a lot of ChIP-seq data from published studies. This is another example of one of our earlier studies. So as we mentioned in here, depending on how you do the ChIP-seq, if you do sonication, the fragments are usually longer. You will probably get, a fragment that cover, 2 to 3 nucleosomes at a time. And so overall, you will see a broad peak. Maybe the middle is slightly lower, but overall, you will see kind of a strong peak. But if you use sonication to get mononucleosome and then sequence, what you will see is that there's one peak in here, there's another peak in here, and the middle will be actually kind of lower signal, and you can actually get a good resolution of the signals. And so this is one example of a study where we look at the K27 acetylation or K4 dimethylation of small intestine. So in small intestine, the crypts here at the cells at the bottom of the small intestine, they have the stem cells, and they continue to grow new cells or proliferate. But then the differentiated cells will be pushed to the top of the villi. And these cells, grow, they differentiate, and they have more absorption. So the food is kind of in here, and these cells absorb the nutrient into inside the villi and to be transferred to the blood to the rest of your body. And so you can see here the yellow kind of progenitor cells are more stem cell-like, and the cells on the top are more differentiated cells. And in a week time, these cells, these are younger and they become older and older. The older cells eventually in a week's time will be shed into the intestine and will die. And so from the, if you get the small intestine from mice and you just shake them, the villi will be shaked off from the crypt, and you can do epigenetic profiles of these two cell types separately. And what you can see across the genome, there are a lot of locations that the epigenetic signals are very similar between the progenitor cells and differentiated cells. They just say, oh, this is intestine. But then there are some locations like this. In the progenitor cells, you might see 2 well-positioned nucleosomes, and in the middle, there's a nucleosome missing. Whereas in the differentiated cells, they're all kind of weak, 3 weakly marked nucleosomes. And you can imagine this is kind of the priming, very low level of transcription expressed just to mark some enhancers as potentially interesting, but they are not really active. But then during, actually in this case, it's after the the transcription factor is no longer active, this region will kind of have a much weaker mark. But in the progenitor cells, when the transcription factor is really, really active, it binds to the middle and recruits the histone mark writers to write stronger marks to the nearby region. So they raise very strong flags to recruit other transcription factors to bind to this location. And so you can imagine there's like a bunny ear in here, and the middle is where the transcription factor really binds. And if we can identify those locations, again, you can imagine this as a big peak, and this is not a peak, right? Or you can imagine it's like a bunny ear with a trough in the middle. And this actually gives us good resolution to look for motifs. And from this, we can see which transcription factors are really important for the progenitor cell. And in comparison, there are regions in the progenitor cell, they are kind of poised, ready or potentially can be bound. But then in the differentiated cell, when the transcription factor really become active, they bind to some regions like this, and the middle will be much, much lower. And you can look for motifs or do the ChIP-seq overlap. We can also see that, oh, there's another group of transcription factors that are bind to this location. And sometimes you might also see like this transcription factor, it can bind to different locations. Even though it's the same transcription factor, sometimes it uniquely binds to some regions in the progenitor, but in the differentiated cells, it uniquely binds to some other location. This is because the same transcription factor are cooperating with different other transcription factors. So in this case, for example, in the progenitor cells, the CDX transcription factor is working together with GATA. And these two together, they both work as kind of a pioneering factors, and the two together can squeeze out some nucleosomes. And then in the differentiated cells, CDX2 is interacting with HNF4A, is another pioneering factor. And because of the motif location, in other locations, these two are together and they can squeeze out different other nucleosomes. And then they recruit other transcription factors. And then you can see actually they regulate very different genes. In the progenitor cells, it really regulates proliferation, whereas in the differentiated cells, by collaborating with different transcription factors, it regulates a lot of the absorption, nutrient related to the gene functions. Okay, so this is how you can use H3K4 monodiamethylation or K27 acetylation ChIP-seq to help you figure out what transcription factors are important, what transcription factors are binding in this location, and what kind of nearby genes they regulate. Okay, let's see. So a lot of people use histone marks to help them identify the relevant transcription factors to figure out which genes are active or potentially active that can be turned on. There are other histone marks that are also important. So we mentioned H3K4 trimethylation. It marks the promoters and tell us in that cell which type of genes has the potential to be turned on or can be or are already on. Another mark, as we mentioned, is H3K36 trimethylation mark, which tells us the polymerase is elongating along this region and is marking kind of the gene body, especially the exons. And so this is a seminal paper from the Zhang Ren's group in 2009, where they use existing histone marks to look at novel genes in the embryonic stem cell. And so what they saw was, if this region, you can see this region has a K4 trimethylation, and in this region, and then nearby there is K36 trimethylation. That tells us there is a gene in here, because this is the beginning of the gene with K4, and this is the gene body of the gene, which is marked by K36 trimethylation, which means that this gene is going to the left direction, right? There's another example like this. The K4 trimethylation is marking the promoter, and the K36 trimethylation is marking the gene body. So we know there's another gene going in this direction. And interestingly, they see cases when there are regions, people didn't know there are genes in here. But interestingly, we see a very strong K4 trimethylation peak here, and also K36 trimethylation marks nearby that indicate there's a gene previously unknown which is going in this direction, and this probably is marking the potential exons on this gene, right? This is and then they can use RNA-seq later on. Sorry, 2009. This is like before, or at least at the early days of RNA sequencing. When you use microarrays, if you don't have probes, you just don't know this region has genes. But later on, if you use RNA-seq, you can see, indeed, there is a transcript in this location that people didn't know before. And you can use this approach to identify novel genes, especially long non-coding RNA genes in the genome by these molecular beacons, right? There are also genes that are marked by what people call bivalent domain. This is, again, a seminal study from Brad Bernstein's lab, but the figure is coming from a different paper. So we mentioned that in the embryonic stem cells, there are active marks, or at least poised, to be activated genes. They are marked by H3K4 trimethylation. The promoters can also be marked, especially CpG promoters, can be marked by H3K27 trimethylation near the CpG regions if this gene needs to be silent. And so you would imagine a gene is either, promising K4 trimethylation or not promising a K27 trimethylation. But interestingly, in embryonic stem cells, there are thousands of genes that are marked by both K4 trimethylation and K27 trimethylation near the promoter regions or the CpG rich regions in the gene. And when people look at where those genes are, turns out these are very, very important developmental genes, especially transcription factors that need to be turned on later on as the cell differentiate. So somehow the cell knows that these regions are important. They not only have an on switch, they also have an off switch, but just in case that the on and off is not tightly regulated, in order to ensure the on and off are tightly regulated in the embryonic stem cell, before the gene is even turned on, these regions have the on-off kind of a gate, both marked at the promoter region, just so that later on, when the gene needs to be turned on, they can be fine-tuned and really, at the right cell, at the right time, and it's turned on at the right level. And so these bivalent genes turn out is, again, an important beacon to the cell for later differentiation. But human, as a scientist, we are using this to help us understand, these genes are potentially interesting because the cell is telling us that this gene needs both an on and an off gate just to ensure a tighter regulation. And that can tell us which genes are more important. And you can see in here, embryonic stem cells initially is bivalent. It has both K27 acetylation and K4 trimethylation on this nucleosome. But as the cell differentiate, if this factor become really on, what will happen is that eventually this gene will only have K4 trimethylation, K27 acetylation when its gene turn on, and the K27, the off gate, or the K27 methylation mark will be erased. Then this gene will be really on. But if it's off, then the K4 trimethylation mark will be erased. The K27 trimethylation mark is on. Say differentiation, you only need to use this transcription factor once, and then later on it's off. Then they will lose their K4 trimethylation mark and keep the suppressive K27 methylation mark. And eventually they might even have DNA methylation to silence it for even longer. But then the poised ones means that maybe this factor is still useful later in the developmental process. And so they are still in the poised state with the bivalent still available on the cell, on the region. And so this is bivalent gene. We mentioned previously, H3K27 acetylation marks both the active promoter and active enhancer. And the enhancers, they really are enriched near the transcription factor binding. But if you look at across the genome, these are the regular enhancers. It might be bound by one transcription factor, and you can see the histone acetylation mark here. You can see here, this is where the transcription factor binds, and that nucleosome is actually being squeezed out. So on the histone acetylation location, there's like no nucleosome, but the two adjacent nucleosomes are better marked. And then nearby, the signal become weaker and weaker. So if you look at like a bigger peak, this is kind of a few hundred base pair of a peak, but on the nucleosome resolution, you can actually see this trough right in the middle. And this transcription factor binding site is also interacting with other factors such as BRD4, MED1, and P300 in this really sharp region. And you can see here K4 monomethylation also kind of have a similar signal with K4 K27 acetylation, but there are even stronger adjacent noise in here. But interestingly, there are also regions in the genome which have not only strong binding, their K27 acetylation signals are much, much stronger, not in terms of height, but also in terms of the width of the peaks, very broad and high peaks. And so in 2014, Rick Young called those regions as super enhancers, which has very, very strong signals for both K27 acetylation, PO2, MED1, BRD4 signals. Out of these different ChIP-seq, K27 acetylation is the easiest to do. And so now people are using K27 acetylation ChIP-seq to help define those regions that have super strong K27 acetylation signals. Interestingly, the genes, a lot of these super enhancers actually overlap with the beginning of a gene. And the genes that are near these super enhancers turn to have very, very important functions. What Rick Young discovered through later studies was that a lot of the cells, the key, really, really important transcription factors are marked by these super enhancers, which means that their nearby regions have transcription factor binding that induce very, very strong K27 acetylation signals. And so these are called like super enhancer target genes. And these genes are very important for the cell lineage, also for disease, for different disease, especially in cancer. These regions have very, very strong K27 acetylation marks. And the very useful thing is that these signals are more informative than increased gene expression. For example, if you look at gene expression between breast cancer and normal breast epithelial cells, there are many genes that are upregulated. Estrogen receptor is by no means a very, very highly overexpressed genes because in general, transcription factors are not expressed at high levels. But interestingly, when you look at the epigenetics on the K27 acetylation signal, you see huge K27 acetylation peaks near the estrogen receptor gene. And it's a really important sign that estrogen receptor is very important in driving breast cancer development. And so using this K27 acetylation signal, you can see not only we can identify where the enhancers are, what transcription factors might be important, you can also see which regions are these super enhancer regions that indicate that the nearby genes are very, very important, that might be important for differentiation, the cell lineage, and also the disease-dependent genes. It almost indicates that the cell knows these regions need to be highly expressed, and they are using the epigenetic marker. They are like really waving the flags for the transcription factors to bind there to activate the gene to ensure these important genes are highly expressed in the cell. And so these are super enhancer targets. And then in 2015, Wei Li's group also discovered something called super promoters. If you look at the transcription start site of many genes, most of these genes have a strong signal near the transcription start site. Of course, if you look at high resolution, right at the middle of the transcription factor, there might be a little nucleosome-free region because that nucleosome right at the transcription start site are being squeezed out by polymerase 2. And this is kind of a zoom out, 10 kb resolution. So overall, you only see a kind of an overall peak. But interestingly, yeah, so you can see most of these peaks are sharp. Interestingly, for some genes, the K4 trimethylation signals are very broad, like this. So, control genes or these, housekeeping or regular genes are like this. But then there are some genes that are very wide. And some of these are immune-related genes, and others are tumor suppressors, well-known tumor suppressor genes. And just by looking at this, they saw some interesting things. For example, when they look at many K4 trimethylation ChIP-seq data in the public, they can, this is looking at like close to 200 samples. They know these are normal and these are cancer samples that people profiled K4 trimethylations before. And they noticed that, this one gene in the normal samples, you see this gene having like a super promoter, potentially indicating that also, again, is another, flag waving to say that this, tumor suppressors are important. The gene needs to be expressed in order to suppress uncontrollable growth. And for some reason in cancers, the super promoter signals are lost. These become, regular, just a couple kb peaks now. And super promoter genes will have a lower expression. And if you lose a tumor suppressor, the cell actually will start uncontrollable growth and then develop cancer. And so they are using this approach again to help them identify more tumor suppressors in the genome and figure out which genes might be important, right? So if you want to look at more oncogenes that drive cancer development, you do a K27 acetylation ChIP-seq and look for those super enhancer target. But if you want to look for tumor suppressor functions, you do a K4 trimethylation profile, and you look at regions that are super promoters. And so I hope that you can see, depending on the experiment, depending on your biological question, you might do a different histomark ChIP-seq and then give you different insights. So human scientists are using the epigenetic beacons in the cell to also give us biological insights. So we mentioned that histomark is important for telling us what regions are important, including annotating the genes, marking the beginning of the genes, and also finding the enhancers in the genome. One difficulty indeed is one student asked, how do you decide which histomarks to profile? And so this is partially depending on the biological question, partially dependent on the availability of high quality antibodies for that particular histomarks. Ideally, you want some kind of assay that can give you like really high resolution transcription factor binding site. And one assay that was super informative is DNase hypersensitivity profile, or DNase-seq. So DNase is an enzyme that can randomly cut in the genome. In fact, if you have like a naked DNA, you use DNase, eventually it will cut the whole DNA, with enough time, all to single nucleotide, just really shatter the nucleus, the genome into smaller and smaller regions in the genome. But then for this situation, we're using the DNase enzyme to cut not the naked DNA, but to cut the chromatin. Which have different level of like a rubber band, different level of tight or openness. And interestingly, when this happened, the DNase cuts the easy regions first, which are the open chromatins that it was just accessible. You can imagine when you have a rubber ball, the inside is really difficult to get to, but the surface is easier, right? The open regions are easier to cut. And so, and this one, we also don't use a complete digestion. We use a very, very limited digestion. And so it will more cut, by chance, say it only cuts every 12 or 20 kb, but then in these regions that are really open, nucleosome-free regions, histomark locations with K27 acetylation, these are kind of open polymerase 2 regions, which are promoters or transcription start site. And in those regions, it could be by chance DNAs will make 2 cuts in nearby regions that are only, like maybe a few 50 base pair or 200 base pair apart. And so after the DNA's digestion, we ran them through a gel or size selection and only select the short fragments that are, say, less than 300 base pair fragments, and then do high throughput sequencing, or maybe between 50 and 300, because you don't want to sequence the single nucleotide sequence. And then with that, you can run, again, similar to the HistoMark ChIP-seq, you map them into the genome, and you can see this is kind of what the DNA's signal profile look like. Interestingly, if you look at each transcription factor ChIP-seq data, you can see this is from the ENCODE project. They have profiled many different transcription factors together, but if you combine all of their raw reads together to do a combined ChIP-seq density, it really tracks with the DNA's hypersensitivity profiles. And so DNAs really give you a overall high resolution map of all the transcription factor binding sites. And so this is a really fantastic experiment. Another experiment, which is also looking at the chromatin accessibility or open chromatin region, is a later experiment that was developed for about 5 years now by Howard Chang and Will Greenleaf's lab at Stanford. And so this is called ATAC-seq. They use an enzyme called TN5. This is an enzyme that virus use to integrate their transposon or genome into another genome. You probably know that RNA, in order to, some RNA virus would incorporate their genome, convert to DNA and then incorporate their genome into the host genome. And they use this type of transposase to insert their genome into the host genome. And so this is an enzyme that's kind of available. And so it has kind of a DNA in it, or transposon in it. And then it will cut into the genome and insert the transposon region into the host genome. And again, when it does the insertion, it also prefers to cut and insert near the open chromatin region, such as promoters, enhancers. And so it has a similar preference against towards the open chromatin. And the nice thing about this is now, instead of using the transposon, we can give it other DNA sequence. And now scientists discovered we can give it DNA sequencing adapters. And so if you like hybridize the TN5 with the DNA sequencing adapter, it will cut and insert the sequencing adapter into the genome. And after that, all you need to do is to PCR amplify. And so if, say, there are 2 cuts, one in here, one in there, and when you use PCR to amplify, one blue primer, one red primer, it will be able to amplify the sequence in between. And especially PCR amplification also favor the shorter fragments, and so you amplify the 100, 200 base pair fragments out, whereas the cut and the insertion that happen, like 50 kb away, will have smaller chance to be amplified. And so you just PCR amplify them out and then sequence out the shorter fragments. And this experiment is really, really easy to do. And again, you can see here, this is the ATAC-seq profile. This is the DNA's profile. It's quite similar, or approximately similar signals. And so these type of profiles are very useful. Again, similar to a HistoMark ChIP-seq, we do not need a priori any knowledge on what transcription factors are important in that particular cell or tissues. We just do the ATAC-seq profile, chromatin accessibility profile, and use either the motif or other public ChIP-seq to tell us which transcription factors might be important. And again, the chromatin accessibility signal, that the signal peaks actually tell us whether the transcription factor binding is important. The TF binding strength doesn't matter, but the histone mark or the chromatin accessibility binding, like whether it's a 10-fold or 50-fold enrichment, that actually tell us this is having stronger effect. In both the DNase and the TAC-seq, the peaks are enriched in both the promoters. This kind of indicates potential active genes in the cells, and on the enhancers, which can inform us about the relevant transcription factor binding. The advantage of DNase is it's a very comprehensive peaks. The peaks are very, very high resolution. A lot of these peaks are less than 200 base pair in resolution. So for motif finding, for figuring out what transcription factors bind there, it's really super helpful. The difficulty there is this experiment is very difficult to do. If you're not careful, the DNase digestion happen too much, your DNA are all digested into too short fragments. And if you don't digest it well enough, the fragments are too far and it's not enriched near the open chromatin regions. And so a TAC-seq now is becoming more popular because it's very easy to do. Any, like a student can learn and do this experiment and generate really good quality data. And it's also really fast because the TAC-seq protocol and the Illumina sequencing protocol are basically the same experiment. And so you can just do this together. And it requires less starting material. You can start with a few hundred cells, thousands of cells, or tens of thousands of cells. Whereas DNase, usually you will need about 10 to 100 million cells to do. Recently, even single cell TAC-seq technique has been invented, and it's actually working very, very well. So it's really popular. And so in terms of how to analyze this data, again, it's very similar to a HistoMark ChIP-seq. You do the read mapping, you call the peaks, do the QC. You can search for motifs from the peaks, especially near the enhancer regions, if you remove all the annotated transcription start site from reference genome. You can identify the TF family. You can use the DNase or a TAC-seq regulator potential to tell us which genes are potentially active in the cell. And you can also use the TAC-seq or DNA signals to inform how to weight the TF binding on different locations. And you can overlap the DNase and the TAC-seq peaks with ChIP-seq data to find the relevant transcription factors using Systrom Toolkit. Okay, so that's some of the basic analysis. Because there are a lot of existing K27 acetylation, ChIP-seq, and chromatin accessibility profiles already generated in the public, sometimes, even if you only have differential genes without any TF ChIP-seq, HistoMark ChIP-seq, or DNase ChIP-seq, we have an algorithm developed, we call it LISA, that can already tell you which transcription factors might be important by utilizing the public TF ChIP-seq and HistoMark ChIP-seq or DNase data. And so the approach works like this. The algorithm takes an input of say 100 or 200 differential genes between some conditions. And based on those differential genes, the algorithm first look at all the public K27 acetylation or DNase data and use each data to calculate this regulator potential. And so you can see in this case, every column is one DNase or one K27 acetylation ChIP-seq data. And some genes, where we calculate this for every gene, so every gene has a score. And so some genes are high and other genes are low. And these are probably the active genes in the cell. And these are the not active genes in the cell. And so we first ran a regression model to ask if we need to have a kind of we collect, say, the top 10 K27 or DNase profiles using a lasso regression so that the top genes agree with the differential input gene that the query gene list. And so say these 10 are being selected, and we know that using lasso regression, these 10 together will give us the genes, their RPs are very different from the rest based on the K27 or chromatin accessibility profiles. So we say, okay, these are the ones that we select. And then together, these epigenetic profiles, because some of them might be K27 acetylation, some of them might be ATAC-seq or DNase, they tell us which regions are important for enhancers. Then we look at whether it's a TF ChIP-seq in the public or the TF motifs. If we don't have the ChIP-seq data, we ask, okay, which TF ChIP-seq really bind to these enhancer locations that have the K27 signal or the DNase accessibility signals. And then we try to see if you were to not have this TF ChIP-seq or the motif, how much does that change the nearby genes? And we don't just do this in one, we do it across the genome of all these K27 regions or DNase regions. And we ask, how much does it change the nearby gene? And the TF that change as most of the nearby genes as your query gene, that's your TF of interest. And so basically using this, the LISA approach is we first try to use public epigenetic data to select the relevant one that indicate this particular cell type. And using that epigenetic profile overlapping with the TF ChIP-seq or motif occurrence, we can evaluate which of this TF effect has strong influence on the nearby gene expression. And then we'll figure out which factors might be important. And so you can imagine if you compare the differential genes between conditions, like there are some upregulated genes and there are some downregulated genes, you can run this through LISA and it will actually tell you, which transcription factor is important for upregulation, what other transcription factor might be important for downregulation, and there might be some factors that influence both, right? And so you can see here, Cystron Toolkit, if you have just the ATAC-seq data or the DNase data or HistoMark ChIP-seq, Cystron Toolkit can find the TF ChIP-seq that overlap with your ATAC-seq. But if you don't have any of this at all, you only have differential genes, you can still run LISA and use the public ATAC-seq or HistoMark data to help you make predictions. And they both kind of use the TF data in the public as well, which is informative.
Okay, so histomodification is really useful if you don't know the transcription factor that's important a priori, right? You just profile, and then after that you use the motif analysis. Another technology was developed a little later after the histomodification, and it's actually also very, very useful. This is called the chromatin accessibility profile. Initially, people use something called the DNase. So basically, DNase is an enzyme that can just cut the genome. Theoretically, if you have enough DNA, it will chew up the whole piece of DNA into only mononucleotide, really. But if you don't give the chromatin enough of this chewing, you only have a little bit of DNase and a lot of chromatin, DNase will go wherever it's easy to go. But where is it easy? It basically is open chromatin, nucleosome-free regions when transcription factors are there, or the region has a lot of histomarks, the good histomarks that are open. And so DNase will go there and chew. And so if you don't have enough of the enzyme, it only just make occasional cuts. By chance, a lot of the fragments are different. You run this DNase digestion only for a short time, and you run it through a gel, you will see kind of a smear. A lot of fragments are really long and some are a bit shorter, but then in this experiment, you can select the really short fragments that are only maybe 100 or a few hundred base pair long. And this means that this region is somehow very open, that the DNase happen to cut. It make two cuts because one is before and one is after you are selecting the fragment in the middle. And there are two cuts that are fairly close to each other within 100 or 200 base pair apart. And so you can just isolate those short sequence and then sequence them, map them to the genome. And this is again a sequencing experiment from a DNase hypersensitivity experiment. And you can see here, most of the region of the genome is just some random. There are occasional cases that two cuts are close by, but the signal is quite weak. Whereas when in those regions, when transcription factors are binding there, you see very, very strong peaks in there. And in those peaks, you also notice a little bit the plus strand reads are blue, the negative strand reads are yellow, and usually the transcription factor really lands in the middle. Right? So you get very, very good enrichment. And all of those peaks, they tell you transcription factor is binding in those locations. It can be either on the promoter or on the enhancers. And this is a study published by John Stam's group from the ENCODE consortium. And so they did a DNase hypersensitivity assay on this cell. You can see this is the, the roughly region of the chromatin. You can see these are where the peaks are. And it happens that in this cell, scientists have done transcription factor ChIP-seq in many different transcription factors. It's all in the same K562 cell, but the ChIP-seq, this is on the ATF3 transcription factor, this is on the CEBP beta transcription factor, this is on the EGR transcription factor. And when they collapse, basically they added all the reads together from all the transcription factors. See, they created this combined transcription factor density. You can see it overlay really, really well with DNase. And that's really great. It means that if you can do DNase 1 assay, well, potentially you don't need to do, all of those different transcription factors, right? You just do this one, at least these are all the shadows on the screen. The transcription factor only go to those locations, and any transcription factors should bind to this. And that's why if you go back to the previous transcription factor ChIP-seq quality control study lecture, we mentioned that a way to know whether your ChIP-seq data is good quality is you get those peaks and you ask how many of those peaks we have seen in a DNase hypersensitivity study. Most of the time you do a transcription factor ChIP-seq, you get a few thousand peaks, 80 to 90%, or even 98% of the peaks overlap with some DNase before. Because the DNase collected all the, every cell you do a DNase, you get maybe hundreds of thousands of peaks. Another cell you do DNase, you get hundreds of thousands of peaks. You look at all of this, a transcription factor binding should really overlap with all the DNase hypersensitivity peaks. And so having high overlap with the DNase is another indication for quality of your ChIP-seq. Okay, so if you see this region having high DNase hypersensitivity, it means that potentially the transcription factor that can bind there is binding pretty stably and is binding in a lot of cells. Whereas if this region is kind of only a little bit of a peak, that means the transcription factor binding is kind of weaker. It's not in all the cells, in maybe a small percentage of cells. It really can tell you also binding stability, but this stability can be due to the binding of one transcription factor or many, many transcription factor binding all in that location together. Okay, so that's kind of the DNase hypersensitivity peaks. And by the way, for this, you can also look for transcription factor motifs, right? So if you do a DNase, you see what are the differential peaks between a disease state versus a normal state. You get those peaks, you look for motifs, you can see what transcription factor binds there. It's a very important way. So the original study, the authors also want to look at something called the transcription factor footprint. By the way, these peaks, the highest resolution peaks is either the transcription factor itself, because it's only binding by, like a few nucleotide long, or DNase. Histone modification peaks, even the really sharp ones like K27 acetylation, is not as sharp as the DNase peaks. DNase peaks are usually only, 150 to 200 base pair long. It's really sharp, which is really good. But then the developer of this technology, they also want to use something called the DNase footprint to look at transcription factor binding in even higher resolution. And so they look at some locations, for example, if they see a DNase peak in here, but then they really look at not just the peak, they look at where exactly this read lands in this like a 200 base pair location. They zoom in to see where that read lands. And they saw a case where the read is to the left, and there are reads that's really marking the right, but then in the middle, there is a gap where theoretically the transcription factor is binding in there, it's occupying this location, so it's protecting the chromatin from the DNA's cutting. And so you will have a cut here, you might have a cut there, you might be isolating the fragments, you're getting the read from the left and right, but not right in the middle. And so they are using this approach, a footprint, to really look at where the transcription factor really binds, which is really exciting, right? You can use it, you can know the transcription factor binding even to the base pair resolution. The danger of this is the specific way they look at this is they say, if there is DNA signal, if we look at the left end read and the right side, or the read that lands in those locations, if there is a gap in the center, that's an indication there might be some transcription factors binding there. And they specifically have a footprint occupancy score where you can see they look at the C +1, because they don't want to have 0, divided by the reads on the left, and the C +1 divided by the reads on the right. By the way, for L, C, and R, they test between 6 nucleotides to 25 nucleotides. They try everything. And then they ask which one do they get the lowest kind of score, which means that the left and the right is very high, but the center, suddenly you see a trough. That's an indication the transcription factor is binding in the middle, and that creates this footprint. But so one thing we need to be actually very careful about this is DNAs actually have cutting bias. So we look at these properties, even with naked DNA, which doesn't have chromatin. There's no nucleosome, there's no chromatin, it's just double-stranded DNA. You use the MNAs to make the cut. It will still have this distribution, which means that some cuts are more likely to happen in other cuts. And so because with DNAs, every time you see a read, it means there is a cut to right next to it. So we can look at every time the read map there, and we look at the sequence there, and also the 3 nucleotide before this, we can build a cutting model. You can see, for example, some regions can be cut quite frequently, whereas other regions may not be cut at all. And so the intrinsic DNA's cutting bias can have as big a difference as 300-fold, which means that sometimes you might have a left and right, which is very high, and the middle that's really low, but that's caused by a cutting bias. The nucleosome, DNA's enzyme just like to make cuts in here and likes to cut in there, and then in the middle, it couldn't cut very well, and then you would have a gap. And so, so far what we have found is that actually using the footprint to predict the transcription factor is actually not a very good approach. It doesn't really give you better prediction. Instead, if you only just use motif in the DNA's peaks to make prediction, you actually are doing a better job. And so this is a study that was originally published. They say, oh, see, there's a peak in the left, there's a peak in the middle, and there's a gap in the middle. Sorry, a peak in the left and right and a gap in the middle. They say, there's a transcription factor binding there. And they can see it not only in human, they also see the same sequence in mouse having this cutting. There must be a transcription factor binding in this location. It turns out this is actually a bias. If you even look at the genetic DNA, it's because of the DNA. If you just look at the genetic DNA cutting, naked DNA, it also creates this left cut and right cut, and the middle is unfavorable in terms of cutting. And the motif they identify there is not a transcription factor motif at all. So we need to be more careful about this. Okay. Questions about DNA's footprinting. So by the way, the world expert for DNA's hypersensitivity experiment is John Stam's group in University of Washington. He has generated DNA's profile for thousands of different cells in different developmental stage, disease, different cell lines. And his data quality is really, really great. And this data is very, very useful in general, because anytime you are interested in some cell, you can look at his data, at least you know where the shadows are and where you should focus your attention on. Okay, so that's a DNase.
So I hope you can see from the lectures on DNA methylation, the lectures on histone modification, nucleosome positioning, and chromatin accessibility that epigenetics are beacons in the genome that tell transcription factors where to go. And it allows a eukaryote genome, despite having very, very long genomic sequence in the genome, for transcription factors to really quickly find the relevant locations to bind. But once they go there, they kind of help further change the epigenetics. They can also open some regions that were previously not marked. And then they influence the transcription factor, binding, and also the nearby gene expression much faster. And so you can also see that using histone marks, we can annotate, enhancers in the genome, annotate or predict the TF transcription factors based on the chromatin dynamics, find the new genes that are unknown in the genome, identify bivalent genes, super enhancer or super promoter target genes which are having important functions. Chromatin accessibility kind of give us a collection of all the TF as well. You can also use it to identify the driving transcription factors. And so this is kind of like a chicken egg problem. It's, when you have a transcription factor that just become active, the initial binding locations are pretty much dictated by the existing epigenetic profile. Which regions have flags already, that's where the TF will go. But if the transcription factor is expressed for long enough time, you will have some chance to explore some regions that were no mark. You have time to explore those the remaining 3 billion base pair, and then gradually can open up some of those regions, especially if this transcription factor is a pioneering factor. It can open up previously nucleosome bound regions and squeeze out the nucleosome, then recruits the histomark writers to make those nearby nucleosome flagged and for further opening. And so in some sense, the epigenetic mark indicate the effect of all the transcription factors that happen in that situation. And they influence how a new transcription factor would bind. But then that new transcription factor might work with all the existing transcription factors to change the epigenetic landscape over time. And so when the cell from embryonic stem cell to start differentiation, at every stage, the epigenetic will help the new transcription factors to decide where to go, what to express. And the cell will also remember, okay, I'm a blood, I'm not a muscle or a brain cell. And so that's kind of, the epigenetic provides the memory of the cell lineage, but also dictates, what transcription factors might be able to turn on or off. But once the transcription factor is on or off, it kind of helps it move further down the differentiation lineage. And so this is really kind of the chicken and egg relationship. You can also imagine transcription factor expression as the kinetic energy and epigenetics as the potential energy. They kind of influence each other in this differentiation landscape. And so this kind of helps us understand what happens when embryonic stem cell become more and more differentiated as the cell grow into different tissues and organs. And also, the induced pluripotent cell, this IPS process, is from somewhere down the slope, you epigenetically reprogram it to the top of the mountain, to the embryonic stem cells. It's like you are changing the epigenetic status. And in cancer, you can imagine it's like going down ski slopes and suddenly it got stuck in a ditch. That's not really a normal trail, right? So that's actually also an epigenetic, some crazy transcription combination happens and the epigenetic goes in the wrong state and it gets stuck there. And so understanding epigenetics really help us, in understanding gene regulation, differentiation process, disease process, and mechanisms. And so that's very, very important. In the next lecture, we're going to have a swap. We'll first have the hidden Markov model lecture, which is related to chromatin accessibility and histone modification. Then we talk about higher order chromatin interactions. That will conclude module 2. Okay, sorry I'm running over time a little bit, and thank you for your attention.